right, so this is our first lecture, and it is on using the scientific method, which is a topic that most of you should already be familiar with. All right, so the steps of the scientific method in general, as I'm sure you recall, are that we start by asking some kind of a question that we're going to answer. We collect some background information. We make a hypothesis or an educated guess about what we think is going to happen. We perform a controlled experiment. We gather our data and analyze it, and finally, we draw a conclusion which typically is accepting or rejecting the hypothesis that we made. So these are our general steps. So let's start with an example. So let's say that your friend says that listening to music will help you study better. How might we test this? So in class, we discussed some different ideas of experiments that could be done. And what we finally came up with as the standard experiment was, let's just say that we split our class in half, and half of the class is going to listen to music, the other half of the class will not listen to music, and at the end of the week, we're going to take a quiz on the material, and we're going to compare the grades on that quiz of the students who listened to music and the ones who did not. So, hypothesis. So we just said, or your friend thinks, that listening to music is going to help you study better. So what your friend really thinks is that those people who listen to music are going to do better on this quiz that we're talking about. So a hypothesis is our educated guess, and there are two sort of formal ways that we can write our hypothesis. So the first one is our if-then hypothesis. Now, most students have written this before or have heard of this before, but unfortunately, a lot of people think that if it has the word if and the word then, it is a proper if-then hypothesis, and it's not necessarily true. So here's the format. The if part of your if-then is a generality. In other words, what I think, my hypothesis, is really the if. So if listening to music helps me study better, that's generally what I think. That's my generality. My then is the actual experimental results that are going to support it. So if listening to music does help you study better, how are we going to know? The then is that then those students who listened to music while they studied will perform better on the test. So here is a sample. If listening to music helps you study more efficiently, so notice that if I just look at my if right here, listening to music helps me study more efficiently, I already know the whole hypothesis. That is my hypothesis, really. What the then tells me is how we're going to test it. Then, the students who listen to music during their study time will perform better on the weekly test. Aha! So I'm going to have a group of students that listen to music and another group that don't listen to music, and I'm going to give them a weekly test and compare the grades. I now know what the experiment, the general experiment, is going to be. So this would be an appropriate if-then hypothesis. Now, let me show you one that's wrong. If you listen to music, then you will perform better on the test. This would not be a correct if-then, because if I look at the if part, does it tell me what they think? If you listen to music, that really doesn't tell me anything. If I listen to music, no. You notice the correct if listening to music helps you study better, that's what they think. The then part is okay, they'll perform better on the test, but the if part is wrong. So that is an appropriate if-then hypothesis. I will give you another example. Let's say that we were looking at plants and we wanted to see what color of light plants grow best in. We wouldn't say, if you put the plants in red light, they'll grow faster. What you would say is, if red light helps plants grow the best, then the group of plants in red light will be taller than the group of plants in the other colors of light. Now, my if red light helps plants grow better, that tells me what I generally think. Then the group grown in red light will grow taller than the groups in the other colors. I know I'm going to test several colors, and I'm going to look at how tall they are. So it kind of tells me what my experiment is going to be. Okay, the other type of hypothesis, which has become very popular on AP exams, is the null hypothesis. So a null hypothesis may seem kind of silly. because it almost seems like you want your null hypothesis to fail. And you kind of do. You kind of want your null, because we always think of your whole goal in an experiment is for your hypothesis to be correct. Actually, if you write a null hypothesis, the goal of your experiment is that your hypothesis is incorrect, that it's wrong. A null hypothesis is called a no difference hypothesis. It basically says there will be no statistical difference in the grades of the students who listen to music compared to the students who don't listen to music. You could either say it present tense, there is no difference, or I like to sometimes put it future tense, since it's a hypothesis, there will be no statistical difference. Now, that means that if there is a difference, our hypothesis is going to fail, and if there is no difference, our hypothesis will pass. So it kind of seems backwards, and we're going to talk a little more about the statistics later, but I'll give you sort of an overview of why we would want to write it this way. Let's say that we do our test, and let's say that side A, they listen to music, and side B, all the students there, they don't listen to music. Let's say that side A, on the quiz, their average score is an 87. And let's say that side B, their average score is a 64. Most people would conclude, yes, the music made a difference. But what's the cutoff to say music made a difference? For example, if this side got an 87 and this side got an 80, would you say the music helped? Most people would probably still say yes. What if this side got an 87 and this side scored an 85? Would you still say the music helped? In other words, where do we draw the line and say the music made a difference or didn't make a difference? When you use a null hypothesis, we're going to talk next week about statistical analysis. We can put in the numbers, the raw data, and we can say definitively, yes, there was a statistical difference, or no, there was not a statistical difference. In other words, just because 87 is higher than 85, does that really mean the music helped? How much of a difference do we need? Where's our cutoff? So by having a null hypothesis, it sort of sets it up specifically to say, yes, there was a difference, or no, there wasn't. So the statistics part we'll get into next week, but that's a null hypothesis. It's called a no difference hypothesis. You would literally say there will be no statistical difference between the group of students who listen to music and the group that do not on the test. All right, other parts of a controlled experiment. So our independent variable, or our manipulated variable, is the thing we are changing. It's the thing that it's under our control, we control it, and it's the one thing we're changing in the experiment. You should only have one independent variable. In our case, it was whether they were listening to music or not. The music was our independent variable. The experimental group is the group that actually receives the change. So in our case, it would be the group that listened to music. They are receiving music. Now, control group. In the past, you may have just learned control group. That's all I ever learned. However, on the AP exam, they want you to be able to distinguish between a negative control group and a positive control group. So what is that? Well, a negative control group is a group that does not receive the treatment. In fact, I would say the negative control group is probably what you're used to calling a control group. In our experiment, our negative control group is the group that did not listen to music. They did not receive the experimental treatment. If you were testing a drug, the group that didn't get any drug would be your negative control group. So what the heck is a positive control group? Now, not every experiment has both of these. Some experiments only have a negative and some may only have a positive. There should be one or the other. Some experiments may have both. So a positive control group, it sort of shows you that your experiment is working properly. It's a group that's given a different treatment that you know will cause the desired change. Now, that may not make a whole lot of sense. And honestly, I would say for the experiment that we were talking about with music, there really isn't a way we could come up with a good positive control group on that experiment. But here's another experiment where a positive control group does make sense. So let's say we were testing plants. And I mentioned this experiment before, and you wanted to know what wavelength of light made plants grow the best. So we're going to put plants under red light, yellow light, green light, blue light, and we're going to measure how tall they are. A negative control group would be a plant in the dark because it's not receiving light at all. So we're taking away the variable. And what do we expect in that one? We expect basically the plant's not going to grow. But we could also put a positive control group. In other words, how do we know our seeds are good? What if we put the plants under all these different colors of light and nothing happens in any of them? Maybe our seeds are bad. So a positive control group could be a plant in sunlight. In sunlight, we already know, it's established that plants grow well in sunlight. So this would be a good comparison. I'll use sunlight as my positive control group because I know the plants should grow well in sunlight. And then my negative control group will be my plant in the dark, and then my experimental groups will be all my groups of plants that are in the various colors, red, orange, yellow, etc. So that would be an example of a positive control group. Another example of a positive control group would be like when you go to the doctor's office and you take a strep test. So if you've ever thought you had strep throat, here's what the test looks like. You put your sample here, they take a little throat swab, and notice they have a control line and a test line. The control line is technically a positive control group. That line should turn red. It tells us the test works. So even if you don't have strep, the control line should light up. If you do have strep, the control line should light up. If the control line doesn't light up, something's wrong with the test. So a positive control group is something that we know should test positive. It's something that clearly we know does give the results. Controlled variables are the things that would be the same for all of our groups. For example, if we were doing our test scenario, we would probably want to give everybody the same test. They all take the test the same day. We probably want them to listen to the same kind of music. They should have the similar study habits. They should get similar amounts of sleep. That way, if there is a difference in the scores of the kids who listened to music and those who didn't, we can say that it was the music that caused the difference, not that, oh, but group A took the test at a different time of day, or group A, they all listen to different kinds of music, etc. And then our dependent variable is the thing we measured. So in our case, we were going to be actually, it's the data we're collecting. Think of the dependent depends on the change you made. So our data in our experiment would have been the test scores. That's the data we're going to collect. That's our dependent variable. If we were doing fertilizer with plants, our dependent variables might be how tall the plants are. We might measure how many flowers they have. Something like that. But whatever data we collect is our dependent variable. All right, to wrap things up, let's say we're experimenting with a new acne medication. We have 3 groups of 16-year-old males. They all have severe acne. Here's the 3 groups. They're going to receive one of 3 oral medications. Group A is getting a standard treatment of clindamycin. This is a treatment that we know helps with acne. Group B is getting medicine X, the one that we're testing. And Group C is going to get a sugar pill. That way everybody's taking something. At the end of the month, we're going to count how many pimples they have. So let's run through the parts really quick. Our independent variable, what are we changing? We're changing the treatment, which pill they're going to take. Dependent variable, we're going to be counting the amount of acne they have. So dependent, what are we measuring? Independent, what are we changing? This experiment has a positive and a negative control group. Our positive control group would be Group A. They're receiving a treatment that we know already works. Our negative control group is receiving nothing. They're getting a sugar pill. And then our experimental group would be Group B, because we don't know if our treatment works. So again, our positive control group, they're receiving a treatment that we know should work. Group B is our experimental group. We don't know if that'll work or not. And Group C is getting a sugar pill, so this is our negative control group. Controlled variables, they're all 16. They're all males. They all have severe acne. Those are 3 controlled variables. If-then hypothesis. If medicine X improves acne, then the students who receive medicine X should have less acne than those receiving clindamycin or the placebo. So if medicine X works, if medicine X helps with acne, then that group should have less acne than groups A and C. And our null hypothesis, there will be no statistical difference between the number of students with acne in any of the treatment groups. Or there is no statistical difference in the number of, the amount of acne based on the treatment given. Anything like that. So that sort of summarizes the parts of the scientific method. In class, after this, we moved on and we're designing a couple of experiments and we're going to share those tomorrow.